Hello everyone, this is Professor Prit Ashara. I am an assistant professor at Biotechnology Engineering Department of VVP Engineering College, Rajkot. So today we are going to see the basics of bioprocess engineering, what it is, what are the scopes of this branch, and we will be seeing the brief about this branch and the some of the sums or problems that you might face in the upcoming examination of BET, that is BET, and GAT-B, that is GAT-B. So it might help you to recall the concepts. So I will be taking an overview, and as per the engineering curriculum we make this subject study in a full semester. So this will be around 1 hour lecture, and I will try to give as much as basic yet applicable facts of this field as well as some. So let us begin with the concept that uh, as you can see, this is the general diagram that represents the field of bioprocess engineering. So first of all, let us understand what is the bioprocess engineering, which is these integrates the principles of engineering with biological systems to design, develop, and scale up processes for the manufacture of biological products. Let us understand this using the example of insulin. So, if any one of you might be having this information, like previously, for the production of insulin, we were dependent on the pancreas of some of the primitive animals, that is, pigs and such sort of animals. So, what we were doing is we were extracting the insulin out of their pancreas, and we were into so much of exploitation of these animals. But because of the help of the recombinant DNA technology, we could integrate the genome of insulin production into the bacteria, and enhance the production of insulin was shifted to bacteria rather than of animal origin. And it is of much proud for us that Biocon, which is founded and situated at the Bangalore city of India, they are the largest insulin product producers of whole Asia region. So it is really very good thing. But what is the scope of bioprocess engineering into the production of the biological product like insulin? So as you can see in this chart, that is product innovation. So how can we produce the uh, insulin out of bacteria? That is one thing. Then cell line development. So as I just given you the information about the gene integration of insulin production into the bacteria, so we require a particular bacterial cell line through which these will get produced. Then of course bacteria will not survive without their nutritional requirement satisfaction. So what we need to do is we need to give them the basic media components, that is their carbon source, nitrogen source, basic nutrients, minerals, vitamins, etc. So what are the parameters for the process, that is optimized parameters for a particular process? That is what will be the pH of the media, what will be the temperature of the bioreactor, or I can say fermenter, in which bacteria will metabolize or multiply, simultaneously produce the desired product, that is insulin for our case. So these all will be decided, and this is about the upstream processing, my dear students. So how do we define the upstream processing? So there are two technical terms. One is upstream and another is downstream. So what is upstream processing? So whatever steps that we take before starting of fermentation process, or feeding our raw materials, adding our inoculum to the fermenter, all the processes, all the optimization steps fall under the category of upstream processing. So let me break down this for you. So upstream processes is, uh, let us continue understanding the example of insulin production. That is, what which particular gene that I am going to insert into a particular host, which is the location of host, who is the host, which bacterial strain, then integrating of my gene of interest into the host genome, then a selection of fermentation process. Like there are basically three types of fermentation that we will see soon in the subsequent steps of this lecture. So which particular, whether we will be taking batch fermentation, fed batch fermentation, or continuous fermentation, which type, then physical and chemical parameters, like what will be the pH, what will be the temperature of my reaction, what will be the agitation speed, which type of agitators am I going to use. So these all parameters are optimized before taking the production of insulin or any other biological product that we are intending to have, right? So what do I mean by optimizing is, for example, out of 1 liter I am getting 100 grams of final product, or for example insulin. So that is 10% yield. Like for 1000 grams of total feed, what I am getting is 100 grams of final product, which is much higher when you really consider the uh, real time scenario. I mean to say the practical ones. So, but this is, we are just taking an example. So this is a 10% yield. Now, being a bioprocess engineer, and if I am into handling the optimizing the upstream processing, I need to make sure 1000 gram of raw materials are being utilized for producing 100 gram of product with 10% yield. Now, I need to make sure how do I make it, that is final product, from 100 gram to 105, 110, possibly 150 and 200 grams, while feeding 1000 grams of raw material to my bioreactor. Are you getting? So I need to maximize the yield than previously possible. So as we discussed, the first case was of having a yield of 10%. Now we are into maximizing it to 15 or 20% possibly, right? Or else, if we are reducing the uh, amount of raw material, that is from 1000 to 900 or 800, while keeping the yield as 100, so what we are doing is we are increasing the yield only. So in either of the ways, so end goal is minimal media and minimal cost of raw materials, and the optimizing parameters, physicochemical parameters, pH, temperature, and agitation, and all. These should be all so much minimum, and the product should be of higher yield. That is the goal of a bioprocess engineer. And if we specifically focus on upstream processing, then by these fundamentals we can reduce the costing, or that is the role of an upstream engineer. Now comes the fermentation part. So here if you can see, this is a fermenter, and products are synthesized. So bacteria are metabolizing, they are growing, or rather say multiplying, and they are also giving the product. And now we have mixture of products with us. For example, whatever raw material was supplied, whatever bacterial systems, so they are metabolizing, they are dividing. So this is a mixture of what? Bacteria, unutilized raw material, as well as formed final products. All three things are there inside this in the mixed form, right? And these whole mixture we are getting as a final product out of this fermenter or bioreactor, right? So now our goal is to have a very pure or purified insulin that can be readily administered by any diabetic patient to take it subcutaneously via injection. Of course, we cannot supply such products with bacteria. It might harm them. So, how do we purify? How do we purify this mixture to have our product in the finest possible form? And in a way, we are making sure there is nothing except insulin in the final bottle or the my packaging material. So whatever we are doing here in the name of purifying our final product, that can be considered as downstream processing. All right, so let us see the brief about all this. So upstream processing, media components. So we can have a defined media. So all the components of the media are chemically known. For example, if it is made up of 10 different constituents, we know each of them as well as their concentrations. And as compared to that, complex media, which is undefined media, and we do not know the components. It might be present with yeast extract or peptones as well. So these notes particularly I have made from uh, Schuller and Kargi Bioprocess Engineering Principles and Applications book. And these table and whatever figures I have mentioned in this, you can directly go and refer there. You won't find such direct details from the book. Why? Because I have made these notes from the book while referring different chapters and considering the syllabus of GAT-B and BET. So you can consider your superficial notes, I can say, because this is not in detail notes. If you want to refer in detail, means you can go and refer the Schuller and Kargi. You can treat this book as a textbook of bioprocess engineering. We are using it since many years. So don't worry about these parts. These you can confirm and refer for taking more information. So basically this part is from chapter 2, right? Now inoculum, that is the bacterial culture. So age should be age of the inoculum culture strongly affects the lag phase. Now I hope you all know the basic bacterial culture, bacterial growth curve. So it starts with static, then it goes to lag phase, then it goes to log phase, followed by stationary phase, and last uh, you know sort of little bit of decline at last, and which we term that as death phase, right? So this is how your microbial growth curve looks like. And what here condition given is, for the inoculum it should be of lag phase. Why so? Because that is young in age. All right. So once we utilize such sort of young age culture, means whenever we give this inoculum to any culture to get it fermented, means we will get our bacteria adapt log phase soon, and that is what we require in the process. Why? Because in the log phase they multiply as well, and they keep on metabolizing as well. So if we attain stationary phase, there are certain drawbacks, like all the bacteria are into the state wherein they are simply giving us the products. But we, if consider these to be the playground of soldiers, all are strengthful. 100 soldiers are strengthful to combat with 1,000 soldiers of opposition party. But what if I put 1,000 such strengthful soldiers in our team, as we need to compete 1,000 soldiers of opposition team? The war will be completed with short time. Even though previously our team consisting of 100 soldiers were capable of dealing with 1,000 soldiers of opposition team. But now when I am adding of equally strengthful soldiers in our team, means we will win our war very rapidly. Similarly, my dear students, you compare this case with the production, and soldiers you compare it with our bacteria. So if I am having more number of bacteria, means I will get my desired product in a very short span. All right. So this is the beauty of that, and we need to take care of such parameters while we are designing our bioprocess. All right. Now there are different sort of cultures that are utilized. For example, microbial, plant, animal. So any organism which we are intending should be uh, very much known to us. Known to us means what? We should be knowing the metabolic pattern of a particular cell cultures. For example, if we are considering microbial cultures, means it will have either bacteria or fungi and yeast, and they are applicable for the production of enzymes, antibiotics, and other metabolites. So whether they will be aerobic, means requiring the presence of air for their metabolism, or they want a complete absence of air, that is anaerobic organisms. Then plant cell cultures, which are used for the production of secondary metabolites and recombinant proteins. And what are the requirements of this culture? Simultaneously, animal cell cultures, which are very sensitive to handle, and uh, specific requirements of handling of these cell cultures, as well as the particular bioreactor designs. These all factors need to be considered, right? Now, as I was telling you, the three types of fermentation process. So one is batch culture, another is fed batch, and continuous. So for a batch culture, cells are grown in a closed system with a fixed initial medium. So there is one bioreactor. You are feeding it with, for example, 100 liters of raw material. You are closing the bioreactor. You are allowing the microbial cells to metabolize, to grow, and then the final whatever mixture you are getting, that is consisting of the bacteria alive, as well as mixed with my intended final product. And if secondary metabolites are there, then they will also be present in our final mixture. So this whole mixture I will be harvesting at the last, right? So, and yes, this follows a typical growth curve of microbes for a growth, and you don't have any chance to miss any of the phases of the growth for the microbes, right? This is a very important point to consider in the case of batch culture. Now, for the fed batch culture, a little bit of modification is there as compared to the previous one. What is the difference is, nutrients are added intermittently or continuously during the fermentation without removing the culture fluid. So what we are doing is, we are adding some sort of rather I should say very specifically, some nutritional uh, growth is already there. We are allowing bacteria to grow and metabolize. After a short, after a while, we know the dynamics of the process. by what time a particular nutrient will be of deficiency. It can be of vitamins, essential amino acids, or the nutrients, whatever is required, for a particular process to happen, because it is subject to a particular process which we are considering to be uh, the final biological product. So, as per the requirement of the bacterial growth, we at some of the intervals we will add nutritional nutritions externally after some time. For example, after each 3 hours, the new nutrients or fresh nutrients are added to the bioreactor. So it is not a batch culture where I am adding something and keeping it closed for a reaction and then we are harvesting the final mixture. No, what we are doing is at several fixed intervals we are adding some of the nutrients which are required for a process to happen further. This is a fed batch culture. So uh, a reactor is fed with inoculum, the basic media, and all the physicochemical environment. After 3 hours, new nutrients are added, freshly prepared nutrients are added. The next 3 hours again added, and lastly, upon end of the process, we harvest the full mixture. So I hope this is clear, the difference between the batch and the fed batch culture, right? So this why we need to have this particular type of fermentation process to happen, because uh, in the case of batch culture, what will happen is whatever media components are given to the bacteria, that will be kept as it is for the bacteria to utilize. But in the case of fed batch, we are giving the nutrients additionally, and this is keeping our bacterial cells to keep in a particular phase. For example, log phase, we can keep it as long as we want, or as long as it should be there, or as long as it is desired to have a particular phase. And these engineers are very smart and they already know by what uh, amount of time we can stretch a particular phase, and by uh, what conditions we will be in the shift of a particular phase out of the microbial growth curve, right? So this is it for fed batch culture and for the continuous culture. So this is a simultaneous process. What do I mean by simultaneous process? So at the initial phase, a reactor is filled with uh, nutritional values and media components and the inoculum, everything. It is allowed to start the process, and mostly it is getting started in a particular growth phase only. So as soon as the bacteria is added to the media, it starts metabolizing, it starts dividing and start giving us the product. So continuously continuously we are adding the freshly prepared nutritional medium for the bacteria, and some of the part is getting out at the exit, and we are constantly into the process of in and out. What is maintained is chemostat. Chemostat is maintained constant, that is, the chemical constituent or the chemical consistency is maintained for so long time that does not change, or that should not be changing in case of continuous culture, and that is what defines a very good example of continuous culture. uh, because if I can say, if I want to give you a layman example, means uh, you have filled one pot, or I can say a transparent plastic bottle with some of the very fine sand, very small particle sand that is commonly observed nearby beach areas or sea areas, right? So I am adding the water. I am adding the water in a manner that the sand the sand remains as it is. No sand is getting out. If we compare these example with the case of continuous culture, means what? The chemical components are remaining to be constant, and we are removing the other components, that is death phase bacteria, or else you can say the formed product during the fermentation, etc., are retrieved at the last. And what is making these chemical constituents to be stable in the bioreactor is some, that is x grams or x kilograms, are getting out of the bioreactor. Simultaneously, x gram or x kilogram of media components are added to the bioreactor. This is what is keeping our continuous culture to be in the state of chemostat. I hope this is very much clear to you all. Now, Uh, this is an important aspect of considering the growth kinetics, wherein we measure uh, our we measure the growth of microbes in term of time. For example, by what time, from 1 hour from now, or 2 hours, or 3 hours, or 6 hours from the start of a fermentation process, what will be the limiting concentration, uh, uh, of a limiting substrate. This is how we calculate uh, these concentrations. And the formula here given is, let me give firstly brief of this. Microbial growth kinetics describe the rate at which microorganisms grow, and that is we already discussed in the initial part of this lecture. The specific growth rate, that is defined as mu is the rate of increase in biomass per unit of biomass. So 1 unit of biomass is basic biomass. Okay. Now, by what rate, by what rate biomass is increasing, that is what is defined by specific growth rate mu. And monod model is a common unstructured model that relates specific growth rate to the concentration of a limiting substrate. Now, for in the concept of protein and enzyme, it is taught what do we mean by limiting substrate. Limiting substrate, if any one of you don't know, then it is a substrate upon which the reaction will decide whether to proceed further or not. Why so? So, for example, if a process is running out of three substrate, okay, substrate X, substrate Y, and substrate Z, and substrate X is of say for example 30 grams, Y is for 40 grams, and Z is for 50 grams in my initial reaction mixture. Okay. Now, if I want to understand the concept of limiting substrate, means what it will be doing is X to be limiting substrate if the concentration of X, if the concentration of X is deciding the fate of a reaction to proceed further. For example, if my reaction of insulin production is highly dependent on the concentration of substrate X, then my reaction is said to be substrate X limiting. I hope this is clear. Now, to determine this, the formula given is mu equals to mu max multiplied by substrate concentration divided by Ks plus S. Now, mu max is the maximum specific growth rate. Ks is the saturation constant representing the substrate concentration at which growth rate is half of the mu max. Now, I would like to show the figure of this. Just a minute. So this is the graph, as you can see. So why I included, because it makes it easy to see what which values exactly we are seeking, and in case of uh, monode equation or monode model, right? And the next concept is to consider primary and secondary metabolite production. So what are we mean by primary metabolites is which are produced during the exponential growth phase and are directly involved in cell growth and metabolism. So when cells are being divided, or they are growing and metabolizing, whatever production is done by microbes, that we name or that we term it to be primary metabolites. So how then, what about secondary metabolites? They are produced during the stationary phase and are not directly required for growth. For example, if cells are growing, they are growing into the population, they are metabolizing, but that is not the direct product of of the metabolism, of the regular metabolism, specifically, if I can say, right? So I would like to mention here one two examples of the real life. uh, if you are aware about one strain named as chromobacterium violacum, then they produce PHA and PHB, that is polyhydroxy uh, acids, and the precursors of bioplastic. They are the precursor molecules of production of bioplastic. They are widely used, and they are the secondary metabolites of C violacum. And primarily, or I can say primary metabolites are violacin biopigment that they produce on the go, right? Similarly, if you consider Pseudomonas aeruginosa, then they produce pyocyanin, which is again a secondary metabolite. Okay, so we have taken an example, but you may refer this if needed. Now let us understand the basics of downstream processing, which means clarification, separation, purification, and polishing of our final product. So basically, by physical means, we remove particulate matters, primarily cells, other solids from the broth, fermentation broth, separation. For example, filtration sort of technique we apply here. Purification, chromatography, majorly we use under these title, and polishing for our final product stability and formulation up to packaging, right? So these are the unit operations that we do under the level of downstream processing. Filtration, so we have a certain micrometer range of filters to separate the unwanted impurities. Then centrifugation, so usage of centrifugal force to uh, separate the components based on the density gradient or density differences. And precipitation, so if you any of you have performed enzyme purification, means you might be knowing the very basic process named as ammonium salt precipitation, when we wherein we use the concept of keeping or making our mixture rich in salt concentration, which helps a particular enzymes to come down and settle down as precipitates. And this is how we use the method of precipitation to separate particular components out of the mixture, and this makes our purification process really very easy. Chromatography methods, we can spend a whole day discussing different chromatography techniques. Ion exchange is there, into that anionic exchange, cationic exchange chromatography, hydrophobic interaction chromatography, affinity chromatography, size exclusion chromatography. These are all the different type of chromatography techniques which are employed for a specific purposes. For example, our either of the impurities or the final product, if they are charge or ionic charge sensitive, means we can use ionic uh, chromatography, and that too if we know the nature of ions, whether they are cations, whether they are anions. So different types of ions, whatever we know about the nature of our final product or impurity, based on that we can perform a particular purification or separation technique, right? Then this is membrane based methods, which are filtration, different types of filtration, right? Uh, you can refer chapter 11 for this, uh, from Michael Schuler and Kargi book, that is Bioprocessing Engineering Principles and Applications. So let us understand some of the basics of scale up. So till now what we see was the overall overview of the bioproducts, how to formulate, how to design a particular method, what are the factors to be considered, what are the different methods to take the fermentation, what are the ideologies of different fermentation strategies, like batch, fed batch, continuous, etc. So now, when you are designing, a process for 1 liter, for example, I am designing a batch of 1 liter to produce an insulin. All right. Now, of course, we cannot run a multi crore industries through 1 liter of production. I will require crores of different bioreactors to have a good amount of insulin produced and to supply it worldwide, isn't it? Now, what we do is we scale up the process. We need to carry out a fermentation process of say for example 5 liters, 10 liters, 20 liters, 100 liters, 1000 liters, 50,000 liters, 1 lakh liters, likewise. But if you have observed, I have multiplied it by several factors. For example, if I have taken just now, till now, whatever we discussed, you consider that to be an example of 1 liter of bioprocess. Okay. So, what I will be doing is, I will be mixing my inoculum with 1 liter of raw material, or I can say media components, and everything makes up a final volume of 1 liter. Getting? Now, when I want to shift it to the higher scale, that is 10 liters. So, being mathematic is the simple mathematics, I need to simply multiply by 10. No, that is not the right way. Scale up is a different method, or I can say a very different concept and ideology, that is not a direct maths. Like whatever amount you were utilizing in the case of 1 liter, each component should be multiplied by 10, and you are taking a batch of 10 liter. No, never. It happens never. From lab scale to pilot, and from pilot scale to industrial scale, you always face different sort of challenges. It is very important that whatever designing we are doing for a particular scale should be almost equal at each stage. What do I mean is, if I am taking 1 liter of batch, similar results I should be getting in 10 liter of batch, as well as 100 liters of batch, as well as thousands of liters of batch. Production of my particular product or desired product, that is insulin for our case or for our example, whatever metabolites, either primary or secondary, if I am aiming, that should be there present in my uh, final product or final mixture, which need to be purified at last. So each stage should be mimicked when I am thinking or designing a scale up process. I hope this is clear to all, and you may refer these points stated here. Um, these are the different parameters that needs to be considered when you design a scale up process for any biological production for the any biological product, right? So you need to understand what are the power inputs per unit volume of broth, constant KLA, that is oxygen transfer rate. KLA means oxygen transfer rate. That is, there are two terminologies, my dear students, here. One is oxygen transfer rate, another is oxygen uptake rate. Now, when I speak about oxygen transfer rate, when through sparger I supply gas, or for aerobic processes, if I supply oxygen, then from gaseous phase to liquid phase, from gaseous phase to liquid phase, I, I do mean by this is oxygen from gas phase will be dissolved in the liquid phase through phase uh, through phase crossing from gas to liquid. Into liquid it will be dissolved or diluted and will be spreaded homogeneously. This is what we term as oxygen transfer rate. At the rate by which it is getting from gaseous phase to liquid phase, rate at which it is getting dissolved to the liquid phase, or if specifically I can speak, to broth, it is getting dissolved, right? So the rate at which this process is going on, that we name as oxygen transfer rate. And for oxygen uptake rate means for any aerobic processes, each microbial cells would be requiring the presence of oxygen for them to satisfy the metabolic needs. Alright. So the rate by which dissolved oxygen is being utilized by individual microbial cells or plant or animal cells, that is utilizing of dissolved oxygen by cells. The rate by which oxygen is transferred from broth to their cell, that is, my dear students, we name as oxygen uptake rate. I hope this is clear. And to maintain oxygen transfer rate, we need to maintain oxygen transfer rate as well as oxygen uptake rate at some of the synchronicity. What do I mean by synchronicity? For example, the rate at which I am supplying my oxygen to the broth or to the bioreactor is very high, and the oxygen uptake rate, that is utilizing the oxygen by cells, is very slow. That is creating the problem. Why? It is making my process rich, or it is making my broth rich in oxygen, which is not desired. Simultaneously, if oxygen supply rate is very slow to the broth and uptake rate is very high, my dear students, can you assume this is creating a thrust or a back pull wherein bacteria are ready to utilize the oxygen, but oxygen is not present in the enough amount into the broth, isn't it? So we need to maintain this overall oxygen transfer rate, and this is what I mean by KLA, which is taking care of the synchronicity of OUR, oxygen uptake rate, and oxygen transfer rate, OTR. I hope this concept is clear to you all. So there is many more stuff to give you all, but as these being very short intro of bioprocess engineering, let us consider to solve two problems. One related to the growth phase and the kinetics of that, and another related to the growth rate, specific growth rate, and their time, what time we will have our desired population. All right. So I'm reading the problem statement is a batch culture of E. coli is grown on glucose as the sole carbon source. And during the exponential growth phase, the following data is collected. Initial glucose concentration being 20 grams per liter, final glucose concentration after growth 5 grams per liter. So here if somebody asks how much glucose utilized, then we can say 15 grams, isn't it? Now biomass formed for whatever amount of batch is taken, 7.5 grams of biomass is formed, and the product, which is lactic acid, is formed as 3 grams per liter. Based on the data, we need to calculate substrate consumed, that we already calculated. How do we calculate? Initial glucose. So initially this was the concentration. And what was the final glucose concentration? It was 5 grams per liter. So taking the differences, giving us as substrate consumed is 15 grams per liter. This is a very simple mathematics, right? The next we need to calculate is true biomass yield coefficient. So biomass yield coefficient y x by s. So y is yield, x is biomass, s is substrate. Utilize the same to formulate your formula, that is biomass formed divided by substrate consumed. So if I need to if I can explain you, then per unit substrate consumed, per unit substrate consumed, how much biomass is formed. This is what is biomass yield coefficient. I hope this is clear. Now what is given as biomass formed is, this is 7.5. So numerator will be 7.5 and substrate consumed is 15. So 7.5 divided by 15, that is 0.5 grams of biomass. 0.5 grams of biomass is formed, my dear students, per gram of substrate consumed, per gram. So in 1 gram of substrate consumed, 0.5 grams of biomass is formed. So per gram of glucose, half gram of bacteria of E. coli are formed. Getting right? So the next ask this true product yield coefficient, that is y p by s, that is product formed divided by substrate consumed. So denominator remains the same. All right. So what is the amount of product formed? With here it is given, that is 3 grams. So 3 as numerator and substrate consumed we already found here as 15. So 3 by 15 being 0.2. So per gram of substrate, that is 1 gram of substrate, we are having 0.2 grams of product, that is product yield coefficient. All right. So what is the yield of product formation through this reaction is 0.2 grams is produced per 1 gram of substrate, being 20% yield of the reaction. I hope this is clear to all. So very simple mathematics. You can memorize this concept by the coefficient nomenclature here itself. But whatever is there, the reaction is always considered per substrate consumption. So if biomass is asked, means biomass produced per substrate consumption. And if product is asked, you may use product formed divided by substrate consumed. Very easy sum. Alright. Now the next, a microbial culture. You see, this question is almost always asked. Either it is GAT, that is Graduate Aptitude Test for Engineering, that is GATE, or GAT-B or BET. This question is normally found in any life science competitive examinations. All right. So problem statement goes like, a microbial culture has an initial biomass concentration of 0.5 grams per liter. it grows exponentially with specific growth rate. We just saw the concept of mu being specific growth rate, and that is given as 0.35 per hour. What is asked is calculate the doubling time or the time required for the biomass to double. For example, my biomass given is here is 0.5. What will be the time that will be required to convert this into 1 gram per liter? That is, I am doubling the biomass concentration. And the next, it is asked what will be the biomass concentration after 5 hours of exponential growth. So this is the growth rate of 1 hour. What will be after 5 hours? Let us consider calculation. So for the exponential growth, you need to consider these to be final formula, that is x equals to x0 multiplied by e, that is exponent, and to the power mu into t. T represents time, mu is specific growth rate, x is final biomass, this, and x0 being initial biomass. So this is about formula. Now we need to consider time of doubling. So my my final population will be double of the initial. So 2x0, x that is final, is double of initial, that is x0. So now I can write my equation to be, you see here, x equals to 2x0. So here if I put 2x0 means 2x0 equals to x0 into e raised to mu into t. This is what is written here, my dear students. 2 into x0 equals to x0 into e raised to mu t. Now x0 and x0 I can cancel out. So 2 equals to e raised to mu t. If I apply natural logarithm, that is ln, and that is with base e. Always remember, those who don't know, natural logarithm has by default base of e. So if I write ln e base to e, that is 1. So I am applying natural logarithm both the sides. So ln2 this side, that is LHS left hand side, so ln2 equals to ln e base e will be removed and the power will be come into the multiplication. So ln e to the base e will be equal to 1 and rest is mu into t. So my final equation becomes as ln2 equals to mu t. The next step, t equals to ln2 divided by mu, and ln2, if you check in the calculator, that is 0.693 and 0.35, that is specific growth rate that is given here in our problem statement, you see here, right? So this is a simple division calculation. So 0.693 divided by 0.35, around 1.98 hours. So for this biomass, to get double, the time required will be around 2 hours. All right, 2 hours it will take. Now our next question asked is what will be the biomass after 5 hours. So initial is 0.5, right? Initial concentration is 0.5 x0 into this, right? So 0.5 into e raised to 0.35, that is specific growth rate, that is also given here, right? And what is the time? That is in hour 5. So e to the power 0.35 into 5. So e to the power 1.75 multiplied by 0.5. You simply calculate this into your scientific calculator and you get this answer, 2.88 grams per liter. This will be the biomass after 5 hours. So per liter of broth, I will be having 2.88 grams of biomass after 5 hours from start of reaction. I hope this is clear to you all. This was very basic concepts of bioprocess engineering, and we just saw 2 different sums. These can be taken a whole day as well as full semester. Feel free to ask your questions into the comment box. If I can see any of the questions, means I will respond. Thank you so much for your patient listening, and I wish you all the very best for your examinations. Thank you so much.